So we're gonna start in 5 minutes so that people can come and join in. Okay. Just 2 more minutes and we'll start. So we'll start our today's class. I hope everyone is good, they are enjoying this particular course, and you guys are up to date with the with current week's lecture. As we have covered a lot of information, I guess we are in week 7, right? So almost 36 lectures we have covered till now, and you guys have a basic idea about immunology, what are the components, what are the key players, how each component is playing role, and how these components are coming into play and action, right? So again, it is my personal way of learning, and this is what I am implying on you guys as well. So if I continuously go back and revise whatever we have covered, it gives us a good sense of understanding, as well as make us realize how well we have understood particular topic, or whether there are certain confusions or issues associated with that. And carrying on this habit, so what we'll do, we did a whole recap in week 5th, right? So we'll do a whole recap today as well, whatever we have learned, like in a very brief and easy to go manner, okay? And then we'll add few more topics of whatever has been discussed in the week 7, and then we'll go to the questions depending upon the time, right? Either I can stop the revision, or we can directly go to the questions and then again come back and see whatever was covered in week 7, or we can have the revision, and with the flow, with the continuity, we'll cover new topic, and then we'll go to the questions or assignment questions or previous year questions, whatever you want to say, okay? So beginning with this overview, so you know from where we have started, right? So we started with the basic, very basic introduction to immune system. So what it was, so the term immunity usually refers to protection against infectious pathogens. And one more disclaimer, I'm gonna just most likely go through these slides pretty fast unless there are any text, and I'll just pinpoint the major conclusions you should take from these slides, okay? Because we have discussed already twice, so I'm not gonna put too much of time and effort in making you understand the similar thing, okay? Yeah. So the term immunity usually refers to protection against infectious pathogens. However, reactions to some non-infectious substances, including harmless environmental molecules, tumors, and even one's own molecules, are also considered forms of immunity. The collection of cells, tissues, and molecules that mediate these reactions is called the immune system, and the coordinated response of these cells and molecules to pathogen and other substances make up an immune response. So what are the importance of immune system in a human body? Or for us, it is basically they are the defense against the infections, that is their primary function. They are also defense against tumors. They control they also play a role in control of tissue regeneration and scarring, cell injury and pathologic inflammation, as well as recognition of and injury to tissue grafts and newly introduced proteins. So infection and immunity, what are they, and how do they go hand in hand, right? So the most important physiologic function of the immune system is to prevent and eradicate infections. Individuals with defective immune responses are at increased risk for serious, often life-threatening infections. Conversely, stimulating immune responses against microbes through vaccination is the most effective method for protecting individuals against infections. This approach has led to the worldwide eradication of smallpox, and only diseases that has been eliminated from civilization by human intervention. And then we saw the current data, how the diphtheria, measles, mumps, pertussis, polio, rubella, tetanus, hemophilus influenza type B, hepatitis, what their number were in particular years, and how with the help of the knowledge of immunity, immune system and vaccination, we were able to control these diseases. And the live example was COVID-19, which unfortunately all of us have witnessed. Okay, then basically we discussed about the stages of host defense, that is the innate and adaptive immunity. Now from here we'll be dividing the immunity or immune response in two different phases. First will be the innate and second will be the adaptive immunity. So the defense against infections is provided by the early reactions of innate immunity and the later more powerful reactions of adaptive immunity. Innate immunity, also called natural or native immunity, it is present in all healthy individuals at all times. First line of defense blocks microbial entry and eliminate invaders rapidly, provides immediate response upon infection, does not require prior exposure to pathogens, evolutionary older defense mechanism. Then what about adaptive immunity or specific or acquired immunity? Activated after exposure to microbes, requires proliferation and differentiation of lymphocytes. Highly specific to the invading pathogen, involves immunologic memory, stronger response and re-exposure, takes few days to develop, and more specialized and potent due to clonal expansion and effector diversity. So now when we are going back to the from where we have started, now you guys have a better understanding of these terminologies, right? What is clonal expansion, how adaptive immunity is only activated once it sees the microbe, right? We have already covered most of these points on individual basis or individual classes. So now it will give you a very bird's eye view of the whole subject. Okay, then we delve into the components of innate immunity and adaptive immunity. So the innate immunity have epithelial barriers, mast cells, phagocytes, complements, NK cells and dendritic cells. Okay, and in adaptive immunity we have lymphocytes, that is B lymphocyte and T lymphocyte, which ultimately leads to antibodies, plasma cell, that is the memory cell, and effector T cells. Okay, now we'll take each part of immunity, that is innate and adaptive, separately. So once we start with the components of innate immunity, so it is the first line of defense: epithelial barriers, skin and mucosal tissue, antimicrobial substances from the epithelial cells, resident immune cells in or under epithelia. Their major function is to block the microbial entry. Then second line of defense, that is even if the first line of defense doesn't work, then what will be the first thing the microbe will encounter? So they will have phagocytic cells such as neutrophils and macrophages. They'll have plasma proteins such as complement system, and their major function, the second line of defense, the function includes destroying microbes in tissue or circulation. Then additional role of innate immunity is that it triggers or informs the adaptive immunity or adaptive immune response, recognizes limited shared microbial structures, that is PAMs. The adaptive immune response is mediated by lymphocytes with highly diverse and variable receptors for foreign substances and the products of these cells such as antibodies and other proteins. Adaptive immunity, the key features of adaptive immunity includes essential for defense against pathogens that evade innate immunity. Lymphocytes express specific receptors to detect microbial and non-microbial molecules. Now you are aware of what we are talking about here. We are talking about the processed peptide, which will be the microbial peptide, and the non-microbial molecule, that will be the MHC molecules on the APC cells. Okay. It recognizes a wide range of antigens. Antigens, they are basically any molecule specifically which recognized by the lymphocytes or antibodies and is able to elicit an immune response. Now when we talk about adaptive immunity, even adaptive immunity itself can be further divided into two major ways, like based on where it is acting. So one will be the humoral immunity and another will be the cell mediated immunity. So there are two types of adaptive immunity called humoral immunity and cell mediated immunity. They are mediated by different cells and molecules that provide defense against microbes in different locations. Extracellular microbes, which survive outside host cells and are readily destroyed when they are ingested by phagocytes, are combated by antibodies. Microbes that have evolved to survive inside host cells, either in phagocytic vesicles or in the cytosol, are eradicated by the actions of T lymphocytes. Now you know what we are talking about. Here we are talking about antibodies, which are the secretory B cell receptors, and here we are talking about T lymphocytes, specifically CD8 positive or cytotoxic T cell. So when we look at the humoral immunity, in humoral immunity, the responding lymphocyte is B lymphocyte, and the effector mechanism is basically by secreting the antibodies. And what these antibodies will do, these antibodies will prevent infection and eliminate extracellular microbe. Okay, when we talk about cell mediated immunity, in cell mediated immunity, the pathogen is either inside the cell, and inside the cell it can be found in two places. Either it can be in a phagocytic vesicles, or it can be in the cytoplasm. So if the pathogen is in the vesicle, then the responding lymphocyte is helper T cell. It basically secretes certain cytokines on to the cell which is infected, and it leads to the cytokine activated phagocyte killing of the ingested microbe. When the microbe is in the cytoplasm, the responding lymphocyte is cytotoxic T cell, and basically the effector mechanism is directly killing the infected T cell by the cytotoxic T cell, infected microbial cell by the cytotoxic T cell, and then what will be the function? The function is basically killing the cytotoxic T lymphocyte by eliminating the cells, right, directly killing the cell, hence the infection will be removed. Then we delve into a little bit more into the humoral as well as the cell mediated part. So in humoral immunity is mediated by proteins called antibodies, which are produced by cells called B lymphocytes. Secreted antibodies enter the circulation, extracellular tissue fluids, and lumens of mucosal organs such as gastrointestinal and respiratory tracts. Antibodies defend against microbes present in these locations by preventing them from infecting tissue, cells, and by neutralizing toxins made by the microbes. Antibodies also enhance the uptake of extracellular microbes into phagocytes, resulting in the killing of the pathogens. In addition, antibodies are transported through the placenta into the fetal circulation and protect the fetus and newborns from infections. Now you know which particular antibody has the ability to cross the placenta. Not all the antibodies can cross through, right? In cell mediated immunity, so it basically defends against microbes that have entered host cells, mediated by T lymphocytes and not by antibodies. Why? Because antibodies cannot enter inside the cell. Critical for intracellular pathogens, example viruses, bacteria. Limitations of antibodies, obviously because it prevents microbial entry, but ineffective once microbes enter the host cells. Okay. The types of T lymphocytes associated in cell mediated immunity includes helper T cells, that basically produces cytokines, activate phagocytes to destroy ingested microbes in intracellular vesicles, also recruit phagocytes to infection sites to combat extracellular microbes. Cytotoxic T lymphocytes or CTLs, they directly kill infected host cells, including non-phagocytic cells, especially effective against viruses and cytoplasmic pathogens. Immunity may be induced in an individual by infection or vaccination, that is active immunity, or conferred on an individual by transfer of antibodies from an actively immunized individual. It will be termed as passive immunity. Okay. Then if we talk about these two different types of immunities, that is active immunity and passive immunity. Active immunity is developed after exposure to a microbe or its antigens. Individual mounts an immune response and eliminates infection, provides long-lasting resistance, Exposed individual becomes immune. Unexposed individuals are considered naive and they do not have a specific defense against this particular microbe. Passive immunity. Antibodies are transferred from an immune individual or produced in labs. Provides immediate but short-term protection. Does not induce immunologic memory because we are providing the antibodies from the other source. So our own body did not go under clonal selection and making the antibodies and the plasma cells and the effector T cells and the memory T cells. Okay, so hence we don't have any memory of it. What are the examples of such thing is newborns receive maternal antibodies, that is via placenta and via breast milk as well. Clinical applications of these type of immunity, that is passive and active, that is immunodeficiency disorder treated with pooled antibodies. Okay, so even if your system is not able to make antibodies on its own, we can always provide antibodies from outside. Emergency treatment for infections such as snake bites and SARS-CoV-2. Monoclonal antibodies used in cancer immunotherapy. T cell transfer is not feasible between unrelated individuals due to rejection. Now, what are the properties of adaptive immune response? So, few properties of adaptive immune response includes specificity, diversity, memory, and non-reactivity to self. Okay, and now you know about all these properties and how these properties are achieved. We'll shed more light on the non-reactivity to self in today's session. Okay, now this is the clonal selection, the foundation on which we have a very specific immunity against different pathogens. Okay, so as mentioned earlier, there will be a common lymphoid precursor cell. Okay, so if you are talking about clonal selection, here we can see it's about antibodies, so these are B lymphocytes. So lymphocyte clones with diverse receptors arise in generative lymphoid organs. Now you know which different types of B cells and how these receptors are different and how basically these different receptors are made. These cells are present in the lymphoid organ. If we are talking about B cell, then they are in the bone marrow. Okay, now clones of mature lymphocytes specific to many antigens enter lymphoid tissue. Okay, now you can see there are different types of B cells which have different type of B cell receptors and they are lying there. When a microbe enters and the B cell sees, those cells which can bind to the antigen, they are selected and then they are allowed to divide or proliferate in one way. Okay, once these cells are good in number, then they are sent to the place where the infection was arising, and then they basically release the antibody, and the memory cells are also formed in the same process. Okay, now some other features of adaptive immunity, apart from being specific and non-selective to one's own system, so adaptive immune responses have other characteristics that are important for their functions. Immune responses are specialized and different responses are designed to defend best against different kinds of microbes and at different sites of infection. For example, there are several classes of secreted antibodies, each of which performs a different set of functions, and there are several subsets of T cells, each of which combats infection in different ways. Okay, now you know what we are talking about exactly. So we are talking about antibodies, and then we are talking about T helper cells and cytotoxic T cells. All immune responses are self-limited and decline as the infection is eliminated, allowing the system to return to a resting state or the homeostasis, prepared to respond to another infection if it arises. So cells of adaptive immune system includes lymphocytes, that is B lymphocyte and T lymphocyte, and then we have myeloid cells as well, so neutrophils, macrophages, dendritic cells and mast cells. Why these cells are part of adaptive immune system? Because they are the majority of them are the APCs or antigen presenting cells. Okay, now as when we were discussing, I have told you that this is a pretty important graph to understand the whole immune system. So now you can see this from a different light today, because you have already covered majority of the immune system, like how the antibodies work, what is the role of adaptive system. So now if I summarize this, you will be able to better understand and appreciate this graph. So we will see only this purple one. Okay, so we are talking about this. This is an B cell which has receptors against an antigen called X. Okay, so this cell is allowed, this recognized this antigen X and it was allowed to proliferate, and then it secreted the antibody. That is the whole story. But how it actually did? So imagine this antigen X entered on day 0 or day 1. Once this antigen entered, the B cell recognized the PAMs on this antigen X, and then they started to differentiate this particular B cell, and here we are calling it as anti-X B cell. Okay, once these cells are there and divided, so it is taking around 2 to 3 days to elicit an immune response which will reduce the infection level. Okay, so in this case the B cells were divided which had an antigen X type of receptor on them, and then they increased their number and then gradually they decreases because the infection was controlled. Got it? And then the memory B cells remain. So you always have a basic immune response or basic serum level of antibody, indicating that you have different types of antibodies floating in your serum. Now on day 5th or 6th, imagine if you again got the same antigen X, then what will happen? The pre-process was already there. We have memory B cells. So now we don't need to go for clonal selection, then increase the number and then tackle the infection. Now we already have memory B cells which will elicit a high level of immune response by secreting these specific type of antibodies, and then it will curb the infection, and then we'll have again more memory B cells. Okay, the similar trend could be seen for antigen Y. Imagine during the infection or the secondary infection of antigen X, you got the primary infection of antigen Y. So here this will be marked as the first primary infection of Y. So it will follow the same graph of 0th day infection of antigen X. But as it was the secondary response for antigen X, and this will be the primary response for the antigen Y, and the same process will take place for different type of infection. So this is for the simplification purpose. Okay, at one time our body is tackling multiple types of infections. So these graphs will be overlapping if you just take maybe 3 antigens, 10 antigens. More the number of antigens you will see, more closer these graphs will be showing their primary and secondary responses. Okay, then we talked about the basic lymphocytes or types of lymphocytes. So as you know, B lymphocyte, they are the ones which secrete the antibody. Okay, helper T cells, these are the ones which basically help the APCs in controlling the infection. CTL or cytotoxic T cells, these are the ones which directly kill the infected cell. When we talk about regulatory T lymphocytes, these are the ones which maintain the number of B cell and T cell, majorly T cell, to regulate the total number of B cells and T cells into the system. Okay, and to culminate this whole discussion, this very beautiful animatory video was there. So we'll just skip to the major part, right? So So now you can see how revising things at equal intervals of time help us in better understanding the topic, right? So whatever was new to you at some point of time, these things are now clear because you have gone through the subject and you know what are the key players in the immunology.
Despite being surrounded by harmful organisms, toxins, and the threat of our own cells turning into tumor cells, humans manage to survive, thanks largely to our immune system. The immune system is made up of organs, tissues, cells, and molecules that all work together to generate an immune response that protects us from microorganisms, removes toxins, and destroys tumor cells. Hopefully, though, not all at once. The immune response can identify a threat, mount an attack, eliminate a pathogen, and develop mechanisms to remember the offender in case you encounter it again, all within 10 days. In some cases, like if the pathogen is particularly stubborn, or if the immune system starts attacking something it shouldn't, like your own tissue, it can last much longer, for months to years, and that leads to chronic inflammation. Your immune system is like the military, with two main branches: the innate immune response and the adaptive immune response. The innate immune response includes cells that are nonspecific, meaning that although they distinguish an invader from a human cell, they don't distinguish one invader from another invader. The innate response is also feverishly fast, working within minutes to hours. Get it? Feverishly? That's because it's responsible for causing fevers. The trade-off for that speed is that there's no memory associated with innate responses. In other words, the innate response will respond to the same pathogen in the exact same way, no matter how many times it sees the pathogen. The innate immune response includes things that you might not even think of as being part of the immune system. Things like chemical barriers, like lysozymes in the tears, and a low pH in the stomach, as well as physical barriers, like the epithelium in the skin and gut, and the cilia that line the airways to keep invaders out. In contrast, the adaptive immune response is highly specific for each invader. The cells of the adaptive immune response have receptors that differentiate one pathogen from another by their unique parts, called antigens. Adaptive immunity is also diverse, meaning it can recognize almost an infinite number of specific antigens and mount a specific response against each of them. The trade-off is that the adaptive response relies on cells being primed or activated so they can fully differentiate into the right kind of fighter to kill that pathogen, and that can take a few weeks. But the great advantage of the adaptive immune response is immunologic memory. The cells that are activated in the adaptive immune response undergo clonal expansion, which means that they massively proliferate. And each time the adaptive cells see that same pathogen, they massively proliferate again, resulting in a stronger and faster response each time that pathogen comes around. Once that pathogen is destroyed, most of the clonally expanded cells die off, and that's called clonal deletion. But some of the clonally expanded cells live on as memory cells, and they're ready to expand once more if the pathogen ever resurfaces. Now it's time to meet the soldiers, which are the white blood cells or leukocytes. Hematopoiesis is the process of forming white blood cells, as well as red blood cells and platelets, and it primarily takes place in the bone marrow. Hematopoiesis starts with a multipotent hematopoietic stem cell, which can develop into various cell types. Its future is undecided. Some become myeloid progenitor cells, whereas others become lymphoid progenitor cells. The myeloid progenitor cells develop into myeloid cells, which include neutrophils, eosinophils, basophils, mast cells, dendritic cells, macrophages, and monocytes, all of which are part of the innate immune response and can be found in the blood as well as in the tissues. The neutrophils, eosinophils, and basophils are considered granulocytes because they contain granules in their cytoplasm. And neutrophils in particular are also referred to as polymorphonuclear cells, or PMNs, because their nuclei contain multiple lobes instead of being round. During an immune response, the bone marrow produces lots of cells, many of which are neutrophils. Neutrophils use a process called phagocytosis. That's where they get near a pathogen and reach around it with their cytoplasm to swallow it whole, so that it ends up in a phagosome. From there, the neutrophils can destroy the pathogen using two methods. They can use their cytoplasmic granules, or oxidative burst. First, the cytoplasmic granules fuse with the phagosome to form the phagolysosome. The granules contain molecules that lower the pH of the phagolysosome, making it very acidic, and that kills about 2% of the pathogens. Now, the neutrophil doesn't stop there. It keeps swallowing up more and more pathogens until it's full of pathogens, and at that point, it unleashes the oxidative burst. During an oxidative burst, the neutrophil produces lots of highly reactive oxygen species like hydrogen peroxide. These molecules start to destroy nearby proteins and nucleic acids within the phagolysosomes, which are the components of the pathogen that has been ingested. The net result is that the pathogen is eliminated. Now, in comparison to neutrophils, eosinophils and basophils are far less common. They both contain granules that contain histamine and other pro-inflammatory molecules. Eosinophils stain pink with the dye eosin, which is where they get their name. They are phagocytic cells, even though it's not their primary mechanism of attack. They are best known for fighting large and unwieldy helminthic parasites, or worms, by releasing molecules that can poke holes in the outer layer of helminths. These cells are also involved in allergic reactions such as atopic dermatitis and allergic rhinitis, also known as hay fever. When involved in allergic reactions, eosinophils degranulate, meaning they release various enzymes and proteins within their granules.
एक दिन किसान के बागान में
अब सबको मिल
Next, you have basophils, and they stain blue with the dye hematoxylin. And unlike neutrophils, basophils are non-phagocytic. On the flip side, they have granules that contain histamine and other pro-inflammatory molecules. Therefore, they are important in initiating allergic responses. Finally, there are the mast cells, which live in tissues, not in the blood, and are very similar to basophils. They are also non-phagocytic and are involved in allergic responses. Next up are the monocytes, macrophages, and dendritic cells, which are also phagocytic cells. They gobble up pathogens, present antigens, and release cytokines, which are tiny molecules that attract other immune cells to the area. Monocytes only circulate in the blood. Some monocytes migrate into tissues and differentiate into macrophages, which remain in tissues and aren't found in the blood. Dendritic cells are the prototypical antigen-presenting cell. Dendritic cells are usually found in sites that are in contact with most external antigens, like the skin epithelium or the gastrointestinal mucosa. When dendritic cells are young and immature, they're excellent at phagocytosis, constantly eating large amounts of protein found in the interstitial fluid. But when a dendritic cell phagocytoses a pathogen, it's a life-changing, coming-of-age moment. Mature dendritic cells will destroy the pathogen and break up its protein into short amino acid chains. Dendritic cells will then move through the lymph to the nearest lymph node, and they'll perform an antigen presentation, which is where they present those amino acid chains, which are antigens, to T cells. Antigen presentation is what connects the innate and adaptive immune systems. Antigen presentation is something that can be done by dendritic cells, macrophages, as well as monocytes, which is why all of these cells are referred to as antigen-presenting cells. Dendritic cells are the best at this process because they are the only cells that live where pathogens enter, through the epithelial like skin, gut, and airways. And they are the only cells that can traffic from these tissues to lymph nodes, where T cells circulate. Now, only T cells with a receptor that can bind to the specific shape of the antigen will be activated, and that's called priming. It's similar to how a lock will only snap open when a key with a very specific shape goes in. However, T cells can only see their antigen if it is presented to them on a silver platter, and on a molecular level, that platter is the major histocompatibility complex, or MHC for short. So the antigen-presenting cell will load the antigen on an MHC molecule and display it to T cells. And when the right T cell comes along, it binds. The final group of blood cells, the lymphocytes, includes B cells, T cells, and natural killer cells. B and T cells make up the adaptive immune response, while natural killer cells are part of the innate immune system. B cells and natural killer cells complete their development where they started, in the bone marrow, whereas some lymphoid progenitor cells migrate to the thymus where they develop into T cells. All of the lymphocytes are able to travel in and out of tissue and the bloodstream. Natural killer cells are large lymphocytes with granules, and they target cells infected with intracellular organisms, like viruses, as well as cells that pose a threat, like cancer cells. Natural killer cells kill their target cells by releasing cytotoxic granules. These granules contain molecules that punch holes in the target cell membrane by binding directly to the phospholipids and creating pores, and release some molecules that get inside the cell and cause target cells to undergo apoptosis, which is a type of programmed cell death. B cells, like T cells, also have a receptor on their surface that allows them to only bind to an antigen that has a very specific shape. The main difference is that B cells do not need antigens to be presented to them on an MHC molecule. They can simply bind to an antigen directly. When a B cell binds to a protein antigen that's on the surface of a pathogen, it's capable of internalizing that antigen, degrading it, and presenting it to T cells. So technically, they're also antigen-presenting cells as well. Like other antigen-presenting cells, the B cell loads the antigen onto an MHC molecule called MHC2 and displays it to T cells. When a T cell gets activated, it helps the B cell mature into a plasma cell, and a plasma cell can secrete lots and lots of antibodies. Typically, it takes a few weeks for antibody levels to peak. The antibodies, or immunoglobulins, have the exact same antigen specificity as the B cell they came from. Antibodies are just the B cell receptor in a secreted form, so they can circulate in the plasma, which is the non-cellular part of blood, attaching to pathogens and tagging them for destruction. Because antibodies aren't bound to cells and float freely in the blood, this is considered humoral immunity, a throwback to the term humors, which refers to body fluids. Now, the final type of lymphoid cell is the T cell, and it's in charge of cell-mediated immunity. T cells are antigen-specific, but they cannot secrete their antigen receptor. A naive T cell can be activated or primed to allow it to turn into a mature T cell by any of the antigen-presenting cells, but most often it's done by a dendritic cell. Now, there are two main types of T cells, CD4 T cells and CD8 T cells, where CD stands for cluster of differentiation. There are hundreds of CD markers in the immune system, and these CD markers are useful in telling different cells apart. For example, all T cells are CD3 positive because CD3 is part of the T cell antigen receptor. So CD4 positive T cells are actually CD3 positive, CD4 positive. And these cells are called helper cells because they're like generals on the battlefield. They secrete cytokines that help coordinate the efforts of macrophages and B cells. Helper T cells can only see their antigen if it's presented on an MHC2 molecule. CD8 positive T cells are CD3 positive, CD8 positive, and they're called cytotoxic T cells because they kill target cells, really similarly to how natural killer cells do it, with one major difference. CD8 positive T cells only kill cells that present a specific antigen on an MHC1 molecule, which is structurally similar to the MHC2 molecule, whereas natural killer cells aren't nearly as specific in who they kill. So now let's go through a complete immune response with a bacterial pathogen in the lungs. To start, the bacteria will have to get breathed in, slip by your nose hairs, past the cilia in the airways, and will then have to penetrate past the epithelium layer of the lungs. Once it's in the lung tissue, the bacteria will start to divide and might encounter a resident macrophage in the lung tissue, which will ingest the bacteria and start releasing cytokines. Those cytokines start the inflammatory process by making blood vessels leaky and attracting nearby eosinophils, basophils, and mast cells, which release their own cytokines and granules, amplifying the inflammation. Neutrophils from the blood, as well as fresh new ones from the bone marrow, dive into the tissue and join the battle. If the pathogen was a virus, Natural killer cells would help destroy the infected cells at this point. This is all part of the innate immune response. Around this point in the infection, immature dendritic cells residing under the epithelium digest the pathogens and move from the lung tissue over to a nearby lymph node where they present the processed antigen on an MHC2 protein to a naive T cell. The dendritic cell, which is part of the innate immune response, bridges the innate and the adaptive immune responses when it presents the antigen to the T cell, which is part of the adaptive immune response. Sometimes, if the infection is spreading, bacteria might find its way to a lymph node without the help of a dendritic cell. In this case, B cells, part of the adaptive immune response, might directly phagocytose the bacteria and present it to a naive CD4 positive T cell. Either way, if the antigen is the right fit for the T cell will begin to differentiate and undergo clonal expansion. Differentiated CD4 positive T cells will release cytokines that will induce B cells to differentiate into plasma cells, which secrete antibodies that will go into the lymph and then into the bloodstream. The antibodies will tag pathogens, making it easier for the phagocytes to eat them. Some cytokines will activate macrophages to kill bacteria that have been phagocytized, but cannot be killed by the macrophage alone unless it gets help from its friends, the T cells, of course. If the pathogen was a virus living and replicating in the cytoplasm of the infected cells, the CD8 positive T cells would kill any infected cells that express the viral antigen on an MHC1. Over time, as the invading pathogen dies off, most of the B and T cells die of neglect, but a few turn into memory B cells and memory T cells, which linger for years in case they're needed in the future. All right, as a quick recap, the immune system has an innate and an adaptive response. The innate immune response is immediate but nonspecific and lacks memory, whereas the adaptive immune response is highly specific and remembers everything, but it takes several days to get started and almost 2 weeks to peak.
Helping current
So why I let the video play, why I let it play full, because now you are seeing every aspect of immune system or immune response. You are aware of the components, you are aware of the details, but as a whole how they are playing role, that story should be very clear in your head. And this comes only when we see things from separating ourselves from that particular topic which we are discussing. So for example, if I'm gonna discuss with you T cell receptors, then you guys will be only remembering the T cell receptor, the structures, the how they are rearranged and all that stuff. But at the end of the day, what is the function of T cell receptor, where they are, how they function? This is a dynamic process, right? So every single cell which we are discussing, or every single receptor which we are discussing, they are playing some roles at the end of the day. And seeing them as a whole, how each receptor, how each cell plays a role in different places of infection when our body is encountering an infection, it helps us to get a better understanding overall. So now every aspect of the visual aid which we just saw, you have read it, you have gone through it, you have understood it. You might not be confident that that the terminology might be off, or say some nitty-gritty details might be off, but overall you have a very good understanding of what is the immune system, what is the immune response, what is innate immunity, what is adaptive immunity. You guys are very well aware of it. Okay, so with this summary, keeping the summary in head, we're gonna go into details of these receptors now. It will be B cell receptor and T cell receptor. Okay, so when we talk about the specificity and receptors of innate immunity and adaptive immunity, now you know what we're gonna discuss. Okay, so when we talk about innate immunity, we have PRRs which identify PAMs. In case of adaptive immunity, we have B cell receptors and T cell receptors. Okay, so in comparison to innate and adaptive, specificity is higher in adaptive immunity and it's comparatively low for innate immunity. When we talk about what sort of microbes they recognize, so innate immunity sees the microbes as whole because they generally phagocytize it, and adaptive immunity sees the processed antigen or the processed microbe, and this is majorly peptide only. Okay, when we talk about receptors in innate immunity, these receptors, the PRRs which I just said, they are called they are of different types, like TLR, toll-like receptor, they are of different types, then RIG, then all those things we're going to discuss in the coming slide. Then if we talk about adaptive immunity, we have immunoglobulins and T cell receptors. When we talk about the distribution, so antibodies, they are surface receptors, right? So they'll be on the surface. When we talk about adaptive immunity, so adaptive immunity comes into play when there is an intracellular pathogen, right? Because if the pathogen is on the surface or not inside the cell and not surviving, we don't need any thing apart from, say, antibody and complement. So complement we have not yet discussed. Complement system in itself is a huge topic and a very well curated defense mechanism of our body. Okay, so we'll discuss it in the coming week. Then if we talk about gene encoding, then you know how recombination happens to make a T cell receptor, B cell receptor, as well as how they are there in the innate immunity, that is the TLRs or RIGs or those kind of things. Okay, now the first thing which we discussed, when a microbe enters and you get inflammation. Inflammation is a type of immune response at the end of the day, right? So it lets the system know that the microbe is here, we are tackling it, and in the meanwhile, whatever we have seen in the visual aid, whatever we have learned till now, those things are happening in the background. Okay, yeah. Oh, hello, I hope I'm audible. Yeah. So when we talk about the inflammatory system, so there are basically 5 to 7 steps in the inflammation. So now, by now you know what inflammation is, but why it is necessary. So we are like a vessel, our body is like a vessel. All our essential organs, all our things, all our cells, all our tissue, all our organ system, they are inside a vessel. So our vessel needs to be impenetrable. But imagine, due to some reasons, that first level of barrier, that is the physiological barrier, is broken and the microbe enters. So you can see that there is a break in the barrier. It can be because of any reason. It can be a wound, it can be a scratch, it can be a mechanical stress, anything, right? So once that barrier is broken, all the things which were there on the surface now have a one-way passageway to our interiors or inside, or like inside the vessel. Okay, so once the microbe enters, okay, so the cells which are there, now you know which cells we are talking about. We are talking about macrophages, we are talking about dendritic cells, we are talking about mast cells, we are talking about all the neutrophils, basophils, which might not be there resident, but small number of them is always present. But to make sure that the infection is curbed, we need to call more of such cells in the place of infection. So this inflammation is a whole process of calling the effector cells on the place where the injury has occurred. Okay, so what are the steps involved in that? So the cells such as macrophages, dendritic cells, they'll be present just below your dermis. Okay, and then once the infection has taken place, they'll recognize it, they'll phagocytize it, and then in the meantime they'll also secrete some cytokines. Okay, once these cytokines are secreted, what it will do, it will allow the blood vessels which are just beneath the surface, it will allow those vessels to be more penetrable, to be not closely packed layers. Okay, this will lead to the invagination or transfer or transportation, whatever term you want to give, transportation of the essential cells. And here, what are those essential cells? They can be neutrophils, they can be basophils, which are there in your blood, to come to the place of infection. That is the whole sole purpose. But cells, they don't have eyes, they don't have legs, they don't have hands. So for example, if there is a passageway, there is a gate, okay, and you, being a human being, you want to go outside that door, what will you do? You'll open the door first, right, with your hands, and then you'll just walk past it, correct? But imagine if you're freely floating in like say swimming pool. If you guys have the experience of swimming, then you know, once you are inside the water, I'm not talking about the surface, okay, once you are inside the water, for you to change the direction becomes difficult in comparison to when you were on the ground, okay. So by training you can do that, but imagine cells, they don't have such trainings, right? They their whole functionality depends upon the signaling, and that signaling is incurred by cytokines in this case. So these were freely floating cells here, okay. What happened when these mediator molecules or cytokines were secreted? Once the cells see a microbe, the passage between this will be broadened, and then these cells, what will happen? These cells now, they have to go out of these gates. So what they'll do, they'll basically let the adhesion molecules which they have attach themselves in the place where there is gap, and then gradually roll out of it and then do their function, okay. This is the major thing and minor thing as well. This is the overall process of inflammation. Here we'll put the cytokines, like which particular cytokine is allowing the blood vessel to broaden, which particular cytokine is allowing the adhesion molecules to be expressed on the surface so the cells can attach themselves, which cytokine is allowing the cells to have a rolling motion. All those things are details, but the overall process of inflammation includes 5 to 7 major steps, which are pretty intuitive, okay. If you see them as freely floating, and they don't have eyes, they don't have legs, but they need to reach a destination. So once you think like this information becomes pretty intuitive, and the similar thing is like displayed here. So the major steps involved in the cell to reach towards the place of infection. So the cell first needs to get attached to the surface, okay. It will start rolling. Once it attaches itself to the surface, then they need to gradually slip from the gaps which were generated and then reach the place where the infection has taken place. So both of these slides, they are pretty intuitive. The thing which you need to learn is, or remember in this particular slide, is which type of adhesion molecule is present where. So selectin is a ligand, okay, for a selectin receptor which is present on the vessels, okay. So selectin and integrin are present on the cell. Integrin receptor or ligand and selectin receptor is present on the vessel. That's it, okay. Now as we are talking about innate immunity, the receptors which are involved in innate immunity are commonly called TLRs or toll-like receptors, okay. Generally they are called PRRs, but major part of PRR is TLR, okay. So in toll-like receptor, they are present in surface, that is on the membrane. Once the membrane is engulfed, they will be present inside the vesicle as well, okay. And in the cytosol, particular type of PRRs such as NOD-like receptor, RIG-like receptor and cytosolic DNA sensor, they are present, okay. There are certain receptors apart from these as well which are present and helps in identifying some common PAMs on the microbes, Okay, now these terminologies are pretty clear to you guys, and hence I'm not elaborating them anymore. Coming to how the macrophage works or functions. So if the receptor recognizes a microbe, the receptor will lead to the signaling. This we have discussed, right? That signaling, which will be the downstream signaling, it will lead to some change or some biochemical change in the cell, so that it can curb the infection. That can be anything. So till now we have discussed how the cell enhances the phagocytosis. This is one way. Another way is that cell will be killed by the cytotoxic T cell. So even the killing of the cytotoxic T cell is a biochemical process, right? Because cytotoxic T cell or CD8 positive T cell doesn't have any like gun or armors, those kind of thing, right? At the end of the day, whatever is happening when we say, oh, inflammation is happening, activation of macrophage is happening, CD8 is targeting, CD4 is targeting, MHC is binding, all these are biochemical processes. Okay, so downstream to the receptor, after the receptor has found its ligand, it can be any ligand, it can be adhesion molecule ligand, it can be toll-like or PRRs meeting PAMs, it can be any receptor which has found its ligand. It will lead to the downstream signaling, and in the downstream signaling mostly it will change the biochemical property, biochemical nature of the cell. These are few which were enlisted here, and more will be there if you refer to any book, or even maybe if I don't remember exactly, but maybe we have particular slide for this as well in our current session. Okay, now if we talk about the antigen recognition by the adaptive immunity, so by innate we have TLRs, in adaptive we have lymphocytic receptors. In lymphocyte we have two types of receptors, B cell receptor and T cell receptor. Okay, so B and T lymphocyte expresses different receptors that recognizes antigen, membrane bound antibodies on B cells and T cell receptors on T lymphocytes. Some questions needed to be answered, we already know what those questions were, and then we directly went towards the antigen receptors of lymphocyte. And here which lymphocytes we are talking about? B lymphocytes and T lymphocyte. So in the membrane form, the B lymphocyte will have a membrane bound as well as secretory type of receptor. For T cell, they won't have, or T lymphocyte, they won't have any secretory form of receptor. Now you know why. Okay, type of antigens which B lymphocyte will identify will be macromolecules such as proteins, polysaccharides, lipids, nucleic acid and small chemicals, while T lymphocytes, they'll only recognize peptides which are part of APC and MHC complex. APC cell which has MHC receptor along with the microbial peptide, this complex will be recognized by the T lymphocyte. Okay, now some very basic one line summary of B cell receptor and T cell receptor. So B cell receptors, they are membrane bound antibodies, recognize diverse chemical structures, proteins, lipids, carbohydrates and nucleic acid, detect native shapes, conformations of macromolecules. Major function include eliminating or binding the microbe and toxin in humoral immunity. T cell receptors recognizes only peptide bound to MHC molecules. Antigens are from intracellular protein or extracellular proteins processed by the APCs. This is important. Any protein, it can be either a internal protein because the microbe is finely processed, or it can be a surface protein, right, which is presented by the APC with MHC molecule. While B cell receptors, they'll only recognize the surface proteins or extracellular things which are shown by the microbe. They do not bind to free or soluble antigens, right? So B cells, they are broad molecular recognition in native form, while T cell, they are restricted because they only recognize the antigen when it is part of the presenting via the APC with the help of MHC molecule. Now if we talk about the structure of antigen receptor molecules, so the domains, here we are dwelling into the structure now, but you know what these receptors are for. Okay, so in domain organization, variable region, the antigen recognition, they differ between lymphocyte clones. Constant region, structurally they are same between different they're conserved along the clones. Okay, sequence variation concentrated in hypervariable region or CDRs, while CDRs, they are of complementary shape to the antigen. We know those loop regions in the antibody, right? They are the ones which are identifying the microbe. What are the functions? So variation limited to small regions minimizes antigen binding diversity while maintaining receptor structure. Okay, when we talk about the like in general how they are associating with invariant proteins, so antigen receptor polypeptides pair with invariant membrane proteins, deliver intracellular signals after antigen recognition. Okay, in complexes, B cell receptor complex includes B cell receptor and other co-stimulatory receptors, while T cell also includes T cell receptor, co-stimulatory receptor, as well as what, MHC receptor or MHC protein. Okay, so in signaling mechanism, because what I have told you, any receptor binding to its ligand will lead to the signaling. Okay, so the antigen binds extracellular portion of receptor, associated signaling proteins phosphorylates on tyrosine residue, initiates signaling cascade leading to the gene transcription, protein production, and then the response. So this was the biochemical change which I was talking about. Once the ligand has made contact with the receptor, a successful contact with the receptor, there will be some change in the receptor which will tell the cell to change its biochemical nature. Here majorly it is via gene transcription, because what are the effector molecules? They are the proteins. How protein is made? Once the gene is transcribed. Okay, now what are the functions of B cell receptor? So in B cell receptor, as you know, we have discussed in the summary as well, that B cell receptor, there are two types. One which is always associated with the membrane, so they are called membrane bound, and major, which is the one which is secreted, and then we call them what? Antibody. Okay, so same as the TCR, B cells or antibody, they also have variable region and constant region, while T cell receptor, already mentioned, they are always membrane bound. They do not secrete, not at any stage that T cell receptors will be secretory in nature. Okay, again, to give this a little more depth, we're not going to complete this particular video. You guys already have this slide, so we are not going to complete it, but just for the visual aid, so that you can see and remember what B cell
machine
and T
learning
cell
course
receptors
is
are. Just excuse me for the ads. Yeah.
For the T cells and B cells, 1. There are two branches, other less. The response consists of humoral mediated
This we have
immunity
already
and
discussed
cell
in
mediated
detail.
immunity.
Okay.
Cells and T cells are both components of the adaptive immune response. They contain infections in different ways. B cells produce and secrete antibodies. Antibodies are proteins that are able to bind to bacteria and viruses. Antibodies binding bacterial cells or viruses prevents infections from spreading. The antibodies block the bacteria from growing colonies, and in the case of viruses, the antibodies bind to the viruses and prevent the viruses from infecting cells. Any disease-causing agent, such as a virus, can be referred to as a pathogen.
The process of antibodies binding a pathogen to contain the infection is known as opsonization. Opsonization by antibodies not only prevents the spread of infection, but marks the bacteria or viruses for destruction. Phagocytic cells circulating in the blood and fluid surrounding the tissues of the body can detect the presence of antibodies bound to pathogens and then engulf and destroy the opsonized bacteria or viruses. These phagocytic cells are not B cells or T cells and are part of the innate immune response. So while the process of opsonization is part of the adaptive immune response, the actual destruction of pathogens involves the innate immune response. Therefore, both branches of the immune system must work together for a complete immune response to infection. Further detail on this phagocytic process is provided and cell-mediated immunity is needed. T cells respond by killing.
Yeah. Okay. So from here onwards, so from here onwards, what we have discussed, we know humoral part involves antibodies. What are antibodies? Antibodies are B cell receptors. Now connect this to how these receptors were made. They were made when clonal selection happened. How did the clonal selection happen? Clonal selection happened when the receptor made interaction with the microbe, or say X microbe. So the cell had anti-X antibody on it, or anti-X receptor on it. How did the cells have anti-X? Because once the B cells are developed in bone marrow, what happens? The selection process happens, or the clonal selection happens. Only the cells which recognize an antigen and then not the self antigen, they are allowed to proliferate. Okay, so from this one term, like antibody, which we just said, you should be able to connect all these things. Okay, here we'll discuss the structure of antibody, because antibodies in itself,
90 of SQL
antibodies in itself are a huge market space for what? For industries to come up with solutions against diseases. So if I am giving antibodies directly to a patient who is suffering from any disease, say, then what type of immunity will it provide? It will be passive adaptive immunity, right? Because the individual in itself is not undergoing the selection, no memory T cells will be formed, only we are supplying the antibodies from the outside. But saying is easier than actually doing it, because there are our immune system, which is, say, it's there for our own health or for our own betterment or for our own survival. It also poses a huge resistance when we just want to transfer one thing from one individual to another individual, right? Then the reaction of anti-antigen, because your antibody will be treated as an antigen in my body, right? Because for my body, your protein is a foreign protein. So how industries are tackling it? So we have already grown through all such things. So vaccinations are there, well prepared antibodies are already there. So it's already there in the market, but still there are many things which we are not aware of, and we are still trying to make better of what is already available. So you can only develop something new when you are pretty clear with the basics and what are the drawbacks of the current technique is available. Then only you can fill the gap, right? So for that purpose, you should be aware of how receptors, specifically B cell receptor or antibody, what is the major components, or what are the major components of antibody, how these components come into play, what are the roles of these components, and whether whole component is important for the functioning, or those n number of questions will be there. So to answer those questions, we're gonna complete this particular video to have a better understanding of antibodies or B cell receptors. Okay.
Plasma B cell and secreted as antibodies into the blood. This is extracellular region of B cell. This is cell membrane. While this is cytoplasmic region of B cell. Immunoglobulins are the glycoproteins, composed of two identical heavy and light chain. The weight of heavy chain is 50,000 to 70,000 Dalton, while the weight of light chain is 25,000 Dalton. There are two types of light chains denoted by kappa and lambda. Cysteine residues form bridge between individual chains of immunoglobulin molecule. This is antigen binding fragment, or Fab, while it is crystallizable fragment, or FC fragment. They are separated by enzyme called as papain. FC fragment possessed binding sites for complement factor C 1q. Light chains consists of two regions approximately of equal size. This is variable region of light chain, subdenoted by VL. And this is variable region of heavy chain, so it is denoted by VH. This is constant region of light chain, so it is denoted by CL. And constant region of heavy chain, so denoted as CH, which is further divided, so denoted as CH1, CH2, and CH3. The constant region varies little from one immunoglobulin to another immunoglobulin. There is huge variability among amino acid sequence in variable region. Both constant and variable region consists of about 110 amino acids. Look at the heavy chain. It has one variable region with 110 amino acid and 3 constant regions. But in case of immunoglobulin M and immunoglobulin E, there are 4 constant domains. Don't worry about the structures of immunoglobulins. They are also explained in detail later in this video. But remember that immunoglobulin M and immunoglobulin E have 4 constant regions. Due to the variability among amino acids, in variable region, immunoglobulin variable portion varies from one immunoglobulin to another immunoglobulin and involve an antigen binding. This is N-terminal. This is C-terminal. Now, C-terminal of B cell receptor has spacer region having 26 amino acids. Then, this is transmembrane portion having 25 amino acids. Then there is short cytoplasmic region, which is very short, having 3 amino acids. Cytoplasmic region of B cell is so small that it cannot activate B cell or cannot give signal to B cell for activation. So, for proper activation of B cell, there are some other components, which is A, heterodimer. Heterodimer means having 2 different monomer. It has one monomer called as Ig alpha, and other is Ig beta. The cytoplasmic portion of Ig alpha and Ig beta have ITAM, or immune receptor tyrosine activation motif. It has amino acids called as tyrosine. This region has kinase enzyme. So this tyrosine is phosphorylated, or phosphate is added to tyrosine through kinase enzyme. Now, signaling starts and leads to B cell activation. This was all about today's session. In next session,
right? So from here, then we delve into the structures of antibody, then the structure of B cell and T cell receptor. So what I'll do is, as I want you guys to have questions done, so what we'll do, we'll start from here in the next class. So maybe next class will take half hour or 45 minutes to completely go through the revision part, and then also come up with the new part which I wanted to discuss with you guys. So that part was basically how T cells mature in the thymus, so which was the topic of week 7. So what we'll do is, we'll try to do some questions, right? We'll try to do some questions, and then we'll complete the revision in the next class along with the new topic. Okay, the procedure is same. I want all of you guys to take participation in the quiz even if you don't know the right answer. For easy questions you guys will get 1 minute, and for difficult questions, depending upon the length, most likely in today's session we don't have many difficult questions. If you have gone through the like week 7 lectures, you won't find any difficult question in today's class. So maybe 2 to 3 minutes for such questions, maybe 1 or 2 question might be there. So we'll start like the previous year assignment with the first question of today. So in radial immunodiffusion, oh no, we have already discussed this, right? Oh yes, my mistake, I'm so sorry. We have to start discussing this. I'm so sorry about that. Yeah, so our first question for today is, nude mice are mice which, first, do not possess bone marrow, do not possess thymus, do not possess B cells, or none of the above. So it's not even a 1 minute question, but alright. So your 1 minute starts now. Feel free to drop your answers in the chat box. Yeah, I don't think so you guys need 1 minute, but let's see. Okay, great guys, I want you. Okay, great, thank you for participation. Then obviously I don't need you guys to give me explanation for this. So the correct answer is that they do not possess any thymus. Okay, so to be very clear with this, because it's a very common question, that what are the difference between skid mice, nude mice. So this is just basic summary of it. Okay, so in nude mice, so what are the origin and genetics of a nude mice? So they carry a spontaneous mutation in the FOXN1 gene, also called the new mutation. This gene is crucial for thymus development. Hence mutation leads to thymic aplasia, okay, which means there is no thymus. While in skid mice, they carry a mutation in the PRKDC gene, that is protein kinase DNA activated catalytic subunit. This gene is essential for DNA repair during VDJ recombination excuse me, which is required to generate functional T cell receptors and B cell receptors, which means nude mice doesn't have functional thymus, while skid mice doesn't have B cell receptor as well as T cell receptor. Okay, so the defects in the immune system for nude mice, they lack thymus, hence there will be no mature T cells, because the maturation of T cells occur in thymus. Okay, B cells are present and can make some antibodies, but antibody responses are impaired because T cell help in the missing, right? Remember, T helper cells, they help B cells. So if there is no thymus, there will be no T helper cell, and hence even the B cell, despite they are present, they won't be functioning at their best. Okay, and natural killer cells are normal. In skid mice, so defective, they are defective in both T cells and B cells. NK cells are intact. Why we are like focusing on NK cells? Because NK cells, they play major role in communicating between the B cell, T cell and the innate immune responses. Okay, so much more severely immunocompromised than nude mice. What are the differences phenotypically? So nude mice, as the name suggests, they are hairless, smooth skin, and they are easy to spot. You can easily see if the mouse doesn't have a fur, they most likely will be nude mice. Sometimes they have other minor defects like eye issues or skin abnormalities as well. While in skid mice, they appear normal, they have a fur, they have coat, they are not hairless. They look like regular mice but are immunologically crippled, which means they don't have any practically existing immune system. Okay, what are the applications of nude and skid mice? So nude mice for tumor xenografts, they used to grow human cancer cells in vivo. Immunological studies, so they study for the functional T cells, transplantation biology, and all that part can be very well studied in nude mice. And the infectious disease research, such as if you want to understand how T cells basically play a role in the infectious disease, you can have a mice model which is nude. Okay, while skid mice, they are majorly used in humanized mouse models. Since both T cell and B cells are absent, human hematopoietic stem cells can be engrafted to reconstitute a human-like immune system, because there will be no rejection, right? Because basically they don't have any immune system to reject the foreign particle. Hence, you can use multiple things which does not require any rejection. Severe immunology studies, such as especially when both arms of adaptive immunity must be absent, widely used in AIDS, leukemia, immunotherapy, and vaccine development research. Still, there are certain limitations. So some limitations of nude mice includes they have B cells and NK cells, right? So can sometimes reject grafts after a while. Not as permissive for xenotransplantations as skid mice, because skid mice doesn't have anything, while nude mice still have B cell functioning, like limited B cell functioning. Skid mice, they are prone to developing leakiness with age. So some T cell and B cell may appear spontaneously due to incomplete defect. Extremely sensitive to pathogens, hence requires strict sterile housing. So the variants and combinations. So genetic engineering has evolved a lot, right? So apart from nude mice and skid mice, you can also combine these type of mice to come up with different types of models. So you can study different phenotypes or different phenomena to extreme levels. Okay, this was a very generic comparison between skid and nude, because most of the students are very confused in this, and you should be very clear what a skid mice is, what a nude mice is. They will also help you in understanding the role of B cells and T cells separately. Okay, so if we summarize the nude mice and the skid mice, so what are the gene defects? So in nude mice, it is FOXN1 gene. In skid mice, it is PRKDC. T cells, so in nude mice, T cells are absent. In skid mice, T cells are also absent. B cells, while in nude mice, B cells will be present, but they won't be functioning to their optimum. In skid mice, there are no B cells. NK cells, in nude mice, they are normal, same as in the skid mice. Hair, in nude mice, as the name suggests, they'll be hairless, while skid mice, they'll have normal hairs, okay, of fur. Immune defects, so nude mice, they are only deficient in T cells because thymus is not there, while skid mice, as there is no B cell T cell, so they are deficient in both T cell and B cell. Uses, you are very well known. Severity, you know severity based upon if there is no immune response at all. So you can study major things with skid mice in comparison to nude mice, and you can clearly see that this is a very normal looking mice, right? So this is a skid mice. So the moment you will put this mice in any open environment where there is a lot of infection going on, these will be the mice which will die first, because they don't have any defense mechanism. On the other hand, this is a nude mice which you can clearly see, and you can identify that this is a nude mice. So this, in comparison to this, they might be able to sustain little bit longer in the environment because they have B cells, but still both of them are immunocompromised at the end of the day. Okay, so the next question for today is, what are double negative T cells? A. T cells that have both CD4 and CD8. T cells that do not have CD4 but have CD8. T cells that do not have CD8 but have CD4. D. T cells that have neither CD4 nor CD8. And your 1 minute starts now. Your 1 minute ends now. So the correct answer is D, that is T cells. Double negative are the T cells that do not have CD4 or CD8 positive. Yes, great. I'll request other people to also take part in answering the questions. So we're gonna see. So if you have gone through week 7, you'll understand this in a far better way, but if not, then you'll get an idea what exactly double positive or such things are.
Do you know what's the future of?
So let me just skip the ad.
Everyone, if you compare the immune system of our body to the defense system of our country, then the T cells of our immune system is very much comparable to the armed military force. They are like soldiers, right? They are like trained soldiers. And in this video, I'll be talking about how T cells develop inside the thymus. But first thing first, before even thinking about what type of training do they get in thymus, we should first talk about how they are born and their birthplace. So the birthplace of T cell is the bone marrow, especially the big bones. Let's say the femur bone. So inside the bone, there would be specific regions. There would be vascular and endosteal niche. And in these specific niche, the T cell or the B cell is born from the progenitor, from the hematopoietic pluripotent stem cell. Now this hematopoietic pluripotent stem cell give rise to lymphoid progenitor or myeloid progenitor. And this lymphoid progenitor is the important one because this gives rise to the adaptive immune system components, whereas the myeloid progenitor gives rise to innate. Lymphoid progenitor in sequential step give rise to T cell precursor. T cell precursor are also progenitor by virtue, and also B cell precursor. Now B cell remains in the bone marrow and their development take place in the bone marrow, whereas the T cell precursor, they leave the bone marrow in order for them to go to a school which is thymus. So they enter the bloodstream and reach the thymus. Thymus is simply the training school of T cell. If we cut a cross section of the thymus, we get a view like that. Now thymus is anatomically subdivided into several regions, and we'll talk about that, its significance. So inside the thymus, there are thymocytes, which are immature T cells. There are also thymic epithelial cells. And the whole thymus is divided into several layers, like subcapsular region, which is the peripheral most region, then cortex, medullocortical junction, and then the medulla. The T cells enter the thymus from the high endothelial venue. Now, once they enter the thymus, they move towards the periphery, and they gather in the subcapsular region. In the subcapsular region, these T cell progenitors can divide and amplify their number. So they really divide and amplify their number in the subcapsular region. From subcapsular region, they descend down to the cortex. And in this process, they encounter thymic epithelial cells, which give them important training. So thymic epithelial cells could be considered as the teachers of these T cells. At this point of time, these T cells are really mature. They would undergo several degrees of training and screening procedures throughout their school. From cortex, they move down to medulla, and they also receive another set of training. And after the medulla, when their training is done, they re-enter the high endothelial venue and migrate around the other parts of the body to the peripheral lymph organs, such as lymph nodes. It takes 3 weeks for this thymic training to complete. So it's a long time for them, right? And next couple of minutes, we'll discuss what type of training do they receive during these 3 weeks. So when the T cell progenitors are in the subcapsular region, they are known as the double negative T cell stage. It's a phase of their life. You can, just like we have our teenage, you can imagine the double negative stage is a phase of the T cell's life. At this point of time, they have the T cell receptor or the precursor of the T cell receptor, but they don't have the co-receptor CD8 or CD4. Now, the T cell receptor could be either TCR alpha or beta, or other, it could be also gamma delta. Now, among these double negative stage, there are substages. And these substages are characterized by specific marker expression. For example, in DN1 stage, c-KIT is pretty high, CD44 is present, whereas CD25 is not present. Whereas if you compare this stage to the DN4 stage, c-KIT and CD45 and CD25, all of these markers goes down. So based on this expression of these markers, you can kind of categorize them into specific phases. Now we'll talk more about these phases in a different video, but for simplicity, let's think about the double negative stage as a unified stage. Now, double negative stage is the time for the VDJ rearrangement of TCR genes. Just like the BCR genes, TCR genes also undergoes rearrangement, and that gives the diversity of the TCR and its ability to interact with so many different peptides.
Shubh, recently I have seen your act where you have
Now, first, the beta chain of the TCR is rearranged, and eventually and sequentially the alpha chain would be rearranged, but they don't get rearranged at the same time. Once both of the chains are rearranged, the mature TCR alpha and beta is formed. But there is also a possibility that it would form TCR gamma and delta. But it turns out that TCR alpha beta is the prevalent isoform over TCR gamma and delta. The reason I'll be discussing it in a different video. But now, after this double negative stage, they descend down to the cortex. They're still in double negative stage. When they go to the cortex, they start expressing both the receptors. Now they are termed as double positive. They started expressing the CD4 co-receptor and the CD8 co-receptor. And already by this time, by the time of double positive stage or at the end of the double negative stage, their TCR gene rearrangement is done and they have a functional TCR by that time. Now, in this double positive stage, they have both the receptors. Now in the cortex, they would first interact with the thymic epithelial cells. Point to be noted that these double positive T cell stage is a prevalent stage present in the thymus. Almost 80% of the cells of the thymus are these DP double positive T cells. Now they would interact with the thymic epithelial cells. The double positive cells has to recognize the MHC bound peptides. So this is the first degree of their training and screening procedure. So their job is to understand peptides that are displayed on the MHC. In later point of their life, they have to do this job. So first, these thymic epithelial cells teach them how to recognize MHCs, be it a class 1 MHC or be it a class 2 MHC. Now, two distinct selection procedure takes place in the cortex. One we call the positive selection. In this selection, the teachers, that means the thymic epithelial cell or the thymus, ensures that these T cells who are double positive, they learn how to recognize MHC bound molecule. So they ensure the MHC restriction. And there is also a procedure of negative selection. In this procedure, they learn how to recognize the MHC molecules, but the high affinity interactions are kind of killed. So this particular negative selection ensures that cell tolerance is maintained and there is no autoreactive T cells generated. In case of many autoimmune diseases, this negative selection mechanism fails, which turns our own defense system against our body, which is detrimental. We will learn about these negative and positive selections slowly and in a lot more details. So first, let's say this particular T cell can recognize the MHC bound peptide. Now these peptides are mostly self-peptides produced by these thymic epithelial cells. In this particular stage, their goal is to get familiarized with the peptides associated with MHC. Now, if these T cells can recognize the MHC bound peptide, then they would proceed further and they would go for the next level of screening. And imagine if they don't or they fail to recognize these MHC bound peptides, they would die by neglect. And it turns out almost 90% of this population is dead because they cannot recognize MHC bound peptide with a decent affinity. So imagine how stringent this process is, right? Only 10% remains and they goes to the next level. The next level is a negative selection. So they had to learn how to select or how to identify MHC bound peptides. But they also make sure that you should not engage with a high affinity interaction. So the T cells which engage with the MHCs with a moderate level of affinity or a low level of affinity, they goes to the next level of selection procedure or they proceed in their career. Otherwise, the T cells which recognize the MHC and with very high affinity interaction, they are dead by the negative selection procedure. They undergo apoptosis. And this is a method of generating tolerance. And at this stage, it is decided that these cells should not survive because if they survive, they would turn against their own body, against our own body. They would create harm in our own body and they can cause autoimmune disorders. So this thymic education is very stringent and that makes sure the T cells gain specific skill sets, but they don't turn against our own body. Now, the job in the cortex is done and they have quite a lot of interaction with the thymic epithelial cells. Now they descend down from the cortex, from the subcorticomedullary junctions to the medulla, where they interact with other thymic cortical cells. And at this stage, the double positive cells either recognize class 2 MHC or they would recognize class 1 MHC. The subpopulation which would recognize class 2 MHC bound peptides, they would eventually stop expressing CD8 and upregulate the expression of CD4. Eventually, they would become the CD4 positive helper T cell. The population that would recognize the MHC class 1 bound peptide, they would downregulate expressing CD4, whereas they would upregulate expression of the CD8. Eventually, they would become CD8 positive cytotoxic T cells. There is also possibility that these T cells that descend down to the medulla, which has already gone negative selection procedure, they don't recognize any of these MHCs or any of these MHC bound peptides. So they would straight cut die by neglect. Now, there are also possibility that they recognize both the classes of MHC. Now, in that situation, they would also die by negative selection because they cannot be non-specific and recognize both class 1 and class 2 MHC bound peptides. So all these different degree of criterias and stringent policies make the T cells really stringent and they learn how to recognize MHCs and MHC bound peptides. Now their training in the thymus is almost at the verge of completion. They would descend down further the medulla, reach the high endothelial venue, and now they would migrate all around the body and they relocate and now they're posted to the peripheral lymph organ like lymph node, which are like army base camp. after these veterans, which are T cells, which got their training, now it's their time to do the field work. In order to do the field work, they have to go to the army base camp, which is the site of action, right? So now they are moving to the site of action, which is the lymph node. And in the lymph node, they reside in specific barracks. There is specific T cell region present in the lymph node. There they encounter dendritic cells and many other cells, and which further help in their differentiation and maturation process. And that I have explained in a separate video called T cell differentiation. I would recommend you to watch.
So this was the major thing which was discussed in week 7. So even if you guys are confused, don't worry, go to the lectures, see it, go to the reference books, and even if that is a trouble, so we are going to discuss the similar thing in the coming week. Okay, so don't go too much into that you couldn't understand a single thing from this. If you have seen the session, you might be able to understand a little bit of it, and if not, just take it as a story. Okay, try to understand what is the role of a T cell receptor. It is a defense system. It should not attack our own system. So what checkpoints this particular T cell needs to pass so that it can do this function? If you think about T cell development in this way, it will be easier. And then these terminologies will come into play. That double negative, double positive, all those things. Okay. So what I'll suggest is we'll meet next in the next class. We'll complete our revision. We'll add the T cell recognition development in thymus. We'll solve more questions based on that. Okay. I hope that sounds good. So thank you so much for your patience, and see you all in the next class. Bye bye.